Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome everyone to the Genetics Podcast. I'm excited to be here today with Dr. Erik Ingelsson, who's the Chief Scientific Officer at Wave Life Sciences, which is a biotech company that you might have heard of a couple of months ago when they announced a world first that we're going to get into. They're focused on unlocking the potential of RNA medicines, and Erik is responsible for growing the company's genetics and genomics capabilities and is really focused on novel, high-impact targets and how you can actually take those from genetic discoveries into having an impact on patients. Prior to Wave, Erik was Senior Vice President of Target Discovery at GSK, and he's also had a number of academic posts, Stanford, Uppsala University in Sweden. So we're going to get into a little bit of the differences between academia, biotech, large pharma. You've seen it all in your career so far. So thank you for joining us today. I'm looking forward to the discussion.
Thanks, Patrick. I'm really looking forward, and thanks for asking me to join.
Yeah, my pleasure. So everybody knows what DNA editing is who listens to this podcast, but RNA editing is, two words we understand, but is a foreign concept actually to a lot of people. I would love to just start there. You could talk a little bit about Wave and give people a sense of the contrasting approaches and advantages and disadvantages of DNA editing and RNA editing.
Yeah, thanks, Patrick. Yeah, so Wave, just to maybe take one step back and then we go into RNA editing. So Wave is a company, and I joined now in May. Really excited about that. We're a company that is built on RNA medicines more broadly. So we use, in contrast to other companies in this space, we use kind of all modalities with a multimodal platform where we, do both. I think many in the audience will be familiar with different kind of silencing approaches. So we use both sRNA, we use ASOs as well for particular, medicines and biological problems. We also have splicing approaches for DMD, for example, and then RNA editing is kind of the last addition to that toolbox. So we have both early and late programs in all of these different things. And RNA editing is, as I said, it's the latest addition. And I think it's a really cool approach where we're basically doing base editing on the RNA level. Whereas with the other approaches, we're using an endogenous enzyme. So for the other, approaches that people may be more familiar with, we use, RNase H, or AGO2 for ASOs and sRNA, respectively. For RNA editing, we use the ADAR enzyme. So it's an endogenous enzyme that we have in our bodies, and it's basically doing editing on the RNA. And what we can do is that we can direct this enzyme to a specific base in the RNA and then do an edit. And you're restricted to certain kind of amino acid substitution, or rather nucleotide substitutions, meaning that you can do certain amino acid substitutions. The main kind of the first application has been to basically do edits of specific mutations. So our first program is for Alpha-1 Antitrypsin Deficiency, where they're driven by a specific mutation that creates this C protein. M protein is the wild type. So it kind of creates this mutated protein. We can, with this approach, revert that back to the functional wild type protein, which is a great approach and really unique approach. And maybe we can get into that later, like how that is different from other therapeutic approaches for AATD. So that's kind of one way of doing it. Now, we have also kind of found out how we can use it for upregulation. So that then unlocks a whole new range of opportunities. Upregulations traditionally, as many of your in the audience will know, is much harder than downregulation, typically in drug discovery. And the way we do that is roughly by doing RNA editing in some of the modulatory regions, like in the promoter region, for example,
Right.
where we have some motifs and we can basically stabilize the transcript and that way increase the levels of the transcript of the protein. So it's kind of a novel approach of using the same RNA editing endogenous ADAR enzyme to kind of just for a different application.
Yeah, that's really
Uh,
interesting. Yeah. If we go, I'd love to actually go into the specifics of the Alpha-1. You
yeah,
talked a little bit about what the small molecule approaches. There are
yeah,
mRNA
hmm
approaches, but
yeah.
yeah, maybe you could tell people about the world first. I think that you published relatively recently, where you actually did RNA editing in humans for the first time.
Yeah, exactly. So we had a proof of mechanism data released now a couple of months ago, probably 2 months ago, where basically after one single dose of our medicine, we saw, and this is just the first few patients, and we had a threshold for like when to disclose that data. And we hit the threshold just after two patients and just at the one single dose, and it's also the lowest
Wow.
dose. So, and this is a low dose. It's 200 milligrams, which is lower than inclisiran, which is 284. So low dose. We have plenty of space actually to go higher now as we move on. We total will be three cohorts with 8 patients in each cohort for a single dose and then multi-dose, so SAD and MAD. This is the first dose, lowest dose, just one single dose, and we already at that point hit kind of the therapeutic levels. So the total AAT levels have traditionally been 11 micromolar. Has been the kind of the goal for the trough level for protein replacement therapies. So after 15 days, after one single dose, we were at that level already. And importantly, more than 50%, so more than 60% actually had edited M protein. So we had about 7 micromolar of M protein. And that is, truly remarkable because these patients, these are CC patients, so they're homozygotes. They don't have any M protein. So any M protein
Wow.
that's produced, it's through the RNA editing mechanism, and we know that. So that was quite astonishing, and we were even like, frankly, a bit surprised it was looking that good already after just
Yeah.
one single dose. So, you know, it really kind of opens up the whole opportunity, both for this medicine. Again, we're going to now proceed with the multiple doses. What our experience from other medicines that we've created is that, the loading adds up being the way our chemistry works is that you get the drug into the organ that in this case is a GalNAc, so we're going for liver, and concentrations kind of go up with multiple doses. So we would expect this to kind of be even more efficient with the multiple doses, and then we even have plenty of space to go higher in dose as well because we're still
Yeah.
at the low dose. So really promising results so far.
Amazing. Yeah, I mean, one of the advantages, but also major disadvantages of DNA editing is the
Hmm.
permanence of it. One of the
Yeah,
advantages, but disadvantage of RNA editing is the impermanence of it.
yeah.
I don't actually really understand the biophysics of it all. Like how often might you
Hmm.
need to dose, and how does it hang around? Because I know it's not one and done, but
Yeah.
it may be not infinite doses
Yeah.
required
So
either.
right, and so we don't know that yet because again, this was actually the first ever RNA editing in human. So I think
Wow.
it's important just to kind of emphasize how big of a deal this was. So we don't really know yet how the preclinical data. We obviously have a lot of like PK data in preclinical models. We don't know yet exactly how that translates into human and how frequent this will be. But based on what we've seen with our medicines that use kind of a lot of the same type of chemistry learnings. We have some unique chemistries. We have something called stereochemistry, where we basically can create stereopure molecules. We have PN chemistry, so that's the substitution of the backbone. And for the RNA editing, we also have 3NU, so one additional edit, like chemical modification. So taking all of those together, we see really good durability and efficiency for our medicines generally. So as an example, we have an obesity medicine, which is an sRNA for inhibin, which is a genetic target, by the way. So maybe we can get into that
Yeah.
later if you want. So that one we do see. Our data indicates that we can get to like once or twice a year dosing. So really infrequent dosing. Now for ADAR and for AATD, that still remains to be seen, but the predictions are pretty infrequent dosing, and whether that's going to end up being bi-monthly, quarterly, every 6 months.
Yeah.
That remains to be seen how that actually translates from the preclinical models into human, because again, this is the first RNA editing medicine.
Is there reason to believe that it would be significantly different? Because you mentioned inclisiran earlier, which I think is the PCSK9
Hmm.
inhibitor, right? And there's a lot of data now to understand
Yeah.
dosing frequency. Is it a similar kind of paradigm there where you can learn from the ASOs and expect similar dynamics in RNA editing?
Yes and no. I would say that, yeah, we can learn that with our chemistry. There are a lot of cross modality learnings in terms of how we build the backbone, and there are a lot of similarities how you build the oligo. Now, but the difference is that it's using a different enzyme. So it's using ADAR instead of for siRNA ACO2.
Right.
It's still going to be potentially different. We can't really say for sure, but we do believe that it's going to be an infrequent dose. So then maybe go back to kind of the differences then from other modalities. I think an important thing is that the current treatment for AATD is really, the currently approved is really just a protein replacement. And AATD has two manifestations. One is lung and one is liver. And the way it works is that, you have these D proteins. They're misfolding due to this mutation, and you get a loss of function in lung. So they get misfolded, they get incorporated or like cluster up in the liver. So that creates a liver disease. That's the gain of function part of the disease. And then you have a loss of function because then it just gets stuck in the liver and it doesn't get
Right.
circulated out and have its function in lung. So these patients experience both lung and liver disease. Now, the current therapy with protein replacement only really deals with the lung disease. So it gives you that protein, so it's a
Mm.
treatment for lung, whereas the liver disease is untreated. Um, there are competitors that are trying with silencing approach, so knocking it down. With siRNA, for example, that would deal with the liver disease. So there are no other
Right.
approaches where you can kind of deal with both problems in one go with one medicine, and also with hopefully infrequent dosing. So I think that's like as compared to other approaches, both current protein replacement and upcoming things in the pipeline where others are trying oligos or silencing approaches. Now, with editing, DNA editing, then, as you mentioned, the big difference is like the permanent nature of that. And uh, that means that there are inherent risks with that. You can't reverse it. That you could have bystander edits, you can have, unintended consequences, and you can't really revert that. So that's the biggest difference, and the biggest problem with it. So I mean, it will ultimately we'll see what patients want and how these processes go. But I think, personally, I would probably prefer to kind of a infrequent dose, it's a subcutaneous
Yes.
medicine. I'd probably rather have that like twice a year or something like that than doing a permanent change with the risks that are inherent. The other thing is delivery. Again, we can our medicine is non-conjugated, so it's basically just an injection, a subcutaneous injections, no conjugates that some other companies are working with, which have their inherent problems. And also, you don't need an AAV or anything like that for delivery.
Yeah.
So it kind of comes with lower risks of adverse effects for that reason as well.
Yeah, it's amazing. This question of one time or once or twice a year. I remember this conversation happening maybe this time last year when
Hmm.
Verve presented some one-time gene editing data, and this discussion was happening around some of the existing PCSK9 therapies
Yeah,
like inclisiran, and would a patient is there patient benefit for having a single one-time compared to a yearly or twice yearly
yeah,
dose? And I think it's exactly the same kind of point, and depending on the disease we may have different answers, but I can
yeah,
imagine that
I
for a
think
lot
so.
of cases what you describe is absolutely right. If we stay in the RNA editing world for a second,
Hmm.
you have you're I think you're planning to apply this to other conditions as
Yeah.
well, like PNPLA3, MASH,
Hmm.
and
Yeah.
some others. Is it a liver focus for the time being, and is that delivery restrictions, or how you think about how this also goes into the lung and brain and all those other important places?
Yeah. So liver has the advantage that we can use the GalNAc conjugate there. So GalNAc, for those that aren't up to speed with what it is, it basically acts as kind of a it's basically glucose type of molecule that derivative that binds to a specific receptor on the liver, or not on the liver, on the hepatocytes. So it's very specific for hepatocytes. So then that gives you kind of a honing missile, getting it incorporated just specifically to the hepatocytes. So for anything where the disease process is in liver, it's a perfect way of dealing with that. Now, we have over the past few years done a lot of work in kind of expanding our toolbox to other organs as well. And by the way, we already have medicines for CNS and muscle as well. So we have a DMD exon skipper that gets really high, industry leading uptake in the muscle. And we have a Huntington allele specific ASO in clinic that again is an intrathecal injection in that case, but we see distribution to the right parts of cortex. So we can get to other tissues. We presented some data at our research day now, maybe a month ago, where we looked at that distribution and where we can get to. And the fascinating thing about this, and my training is I'm an MD and I'm kind of biology background more than chemistry, but our chemists in our company are so amazing, and they basically have built all of these kind of tools for us. This I mentioned this PN chemistry, so it's a backbone modification of a specific part of the backbone where by moving those PNs around and kind of putting them in certain places, and there's some sequence dependency as well, but much is about like how you design that backbone. We can get into different tissues. So we can design something that is specific for fat. We can design something that goes both to liver and fat, and we can design something that goes to muscle. And we're also working on additional tissues, including kidney, lung, and heart as well. So we can get to different things. So with regard to what we have to this point in our publicly disclosed pipeline, it's a lot of liver, as you've noted. We also have programs. We have a program for MSCP2, which is Rett syndrome. That's a CNS program. That's an RNA editing program as well. And then we have a CFTR, so that's cystic fibrosis program, dealing with specific mutations that Vertex drugs can't get to because you get stop codons. So we can, again, using RNA editing, revert that back to healthy wild type as well.
Amazing. I mean, you have for a company of 350 or so people, if my LinkedIn research is correct, it's a remarkable amount of impact and number of programs that you have already. And it sounds like it's a platform you can do so much with. So the question is, where do you focus, not what can we do eventually, right? And maybe that's a good segue into genetics, because that's a key part of the story. You mentioned the INHBE obesity, and maybe love to dive into that as a story that people can learn from, how you think about using genetic targets to inform what you and the team go after.
Yeah, I think it's a great story illustrating this whole concept, and so plus actually links back to kind of my prior life, where I was first leading genetics and later on led target discovery, GSK, because this actually came up through the collaboration between GSK and Wave. Originally, it's a genetic finding that we found as part of that collaboration, but also it was published around the same time by researchers elsewhere, and it comes originally from UK Biobank. So it's it's a predicted loss of function variant. It's heterozygote carriers have a more healthy metabolic profile, so they have a lower waist hip ratio just for BMI. So that reflects like the abdominal obesity. So less of abdominal obesity, lower triglycerides, higher HDL, so the good cholesterol. They have also lower glucose, visceral adiposity as measured with MRI, things like that. They have lower type 2 diabetes risk and lower coronary heart disease risk. Wow. So overall, it's kind of a healthy metabolic profile, and in particular driven by abdominal obesity. So that's kind of what you learn from genetics. And some work that we and others have done is also kind of shown how the biology works. So this inhibin E, or for short, like the full name is actually inhibin beta subunit E. So it's like pretty long. Yeah. So it's basically, it's that transcript goes out. It's transcribed only in the liver. it goes out and creates a homodimer. So two subunits come together, and then it's called activin E. So that gets then circulated. It's a hepatokine. So something that gets released by the liver, goes out. It's a ligand for a receptor in the adipose tissue called ALK7, binds to that receptor, and then blocks lipolysis. So this the function, the natural or the normal function of this hepatokine is to decrease lipolysis, which is the fat burning mechanism in the body. And what then that leads to is increased abdominal obesity, cardiovascular disease, and type 2 diabetes. So that's the normal function. And you could kind of speculate, like, throughout centuries, millennia, how that's been selected for. It's been good to store your energy, right? But in this day and age, it's not a good thing.
Right. We got plenty
So
of it. Yeah.
we have plenty of it. So you don't want to store more energy in your adipocytes. So then these carriers, then they have less of inhibin E, they have less of activin E that releases that brake on lipolysis. It leads to an increased lipolysis. So that's how the genetics points us to us. So that our hypothesis then was that we, with a GalNAc siRNA going directly for the hepatocytes, could decrease levels of inhibin E, activin E, decrease the ALK7 receptor activation on the fat cells, and that should lead to less smaller adipocytes and more lipolysis, and hence all of these good metabolic effects. So that's kind of where this program started. And then we've done a lot of work that we now presented at the research day about a month ago in DIO mouse. So these are diet-induced obesity mice. And interestingly, this all recapitulates like the genetics perfectly, which is always nice. So we do see a reduction of body weight about the same, or actually exactly the same in our model, extent as semaglutide, which is one of the GLP-1s. But importantly, without any effect on the muscle. So one of the kind of problems with GLP-1s, as you may know, is that you get weight loss, but it's kind of both from lean and fat mass. So in this case, and this is, again, same in the human genetics that we see, but also in the mouse, we see no effect on the muscle loss. We do see a visceral fat reduction, which is dramatic, up to 40-50%. And again, on par with semaglutide. We then proceeded to kind of look at, is there an additive effect? So we gave semaglutide plus our inhibin E siRNA, and we doubled the effect. So going from around 10 to around 20. So it seems to have an additive effect. And also importantly, this is just with one single dose, like one dose at the baseline, and then you see a prolonged effect for many weeks. And the third thing that we tried to do was to see, what if we have these mice on a semaglutide, and then we give our inhibin E medicine when they stop taking the semaglutide. So what happens to those that don't get our medicine, the control group, they go back up and they actually overshoot. So they start, being more obese than they were starting with semaglutide. Whereas our medicine kept it flat. So they basically retain their weight gain. So that kind of points us to three different use cases. Monotherapy for individuals that have risk for sarcopenia, such as elderly, for example, those that have a lot of, side effects. GLPs tend to have some GI side effects, for example, and muscle loss. So monotherapy in certain groups. You could also seek like add-on therapy to combine it. And then the third would be that it's a kind of a roadmap to get off of your semaglutide, because that's kind of a general problem for these patients. How do you stop? So in this case, our profile, is, as I mentioned before, it's likely to be once or twice a year. So it's almost like a vaccination for against obesity or something like that, if you want.
Yeah, that's amazing. How I should know this, but is the GLP-1 function well grounded in human genetics or not? Like, what's the mechanism there?
Yeah, there is genetic evidence for it. It didn't come that way. The discoveries weren't made using human genetics, in contrast to PCSK9. This is more of a PCSK9 type of story, this inhibin E, I would say, because it comes from genetics first. So GLPs, no, they were discovered other ways, but however, yes, there is genetic evidence for it. So if you go to big GWASs, there is evidence for that role.
And is it via similar pathway, or are they complete? Because yeah, you're seeing this
Yeah,
independent effect. Because
yeah, there it's
yeah, it's
yeah.
interesting.
Yeah, GLP-1 agonists are primarily through appetite regulation. So they're
Yeah.
centrally acting. And most of other medicines currently in development, like amylin and like other things, they're usually more centrally acting. So this puts this a little bit differently. And it also kind of as illustrated by our results as well, it's more likely because it's orthogonal. This is a peripheral fat mechanism, like
Yeah.
on the abscissa side. So it's more likely to kind of have an additive effect or work in
Right.
tandem. So, and so that's been very exciting. The original genetics finding was done just about 2 years ago, and so we are going into clinic now in Q1. So it's been
Wow.
remarkably fast. We're under 2 years from kind of starting the work until going into the first patient. So
That's
we're
amazing.
filing CTA this quarter, and then we're going into patients in Q1.
Wow. Um, the the transition from GSK to Wave more generally, I mean, you
Mhm.
talked about the maybe these kind of collaborations are a part of this, but
Yeah,
how did you change your approach when you went from a place where you had, there's 5 or 10 large pharma companies where from a genetics perspective, you basically have unlimited resources, right? And then you move
yeah,
into biotech and it's a lot more limited for all the resources that you do
yeah.
have. You have to think differently. What's the biggest shift that you've seen there?
Yeah, I mean, we have to work very differently. Like just for as you pointed out, this is a pretty big difference. Like even like for me, the last organization I led at GSK was like larger than the whole company and
Oh,
at Way. And we have even our own manufacturing, GLP manufacturing in-house. So
wow.
it's quite different in that way. So you have to work much like more agile and smaller teams but you also have to work on the focus. I mean,
Yeah.
with a big company, you could be across a lot of different programs, a lot of different therapy areas. We have to try to stay a little bit more kind of specific on programs
Yeah.
and be on point with them. I think that's a big difference. I think another thing specifically to the genetics part, I think given that we have such a amazing chemistry platform, so I mean I would with the risk of bragging, but I do think we have a world leading oligo platform and RNA medicine platform. That means that kind of opens up the opportunity to work on targets that aren't as novel always. I mean,
Yeah.
actually, some of the targets that we now announced are very well known. Like
Right.
we're the first to, I think, be able to upregulate LDLR. Our preclinical data looks really
Hmm.
kind of promising. People in big pharma have tried with that for decades, and obviously it's a very well-known mechanism, very well-known target. ApoB, same thing. PLM, you mentioned PLMP3 doing that RNA editing, that mutation was, reported originally, I
Yeah.
think, by Helen Hobbs and John Cohen and that group,
Well,
it's
and
more than 10.
Huntington's disease that you
And
mentioned is
hunting, yeah.
about as
We're
old as it gets.
yeah. So I think that's a probably a big difference. Like, so we
Yeah.
have such a kind of a competitive advantage in terms of our chemistry platform that we can kind of get and do. We can do things that others can't do, and we can do it like with better, both going for things that people haven't been able to. So PLMP3, for example, there are plenty of companies that tried with knockdown,
Yeah.
but with sRNA. But the problem is that the wild type has a function. Huntington, same thing. We have an allele specific ASO that knocks down only the mutant but spares the wild type. So kind of the toolbox gives you some opportunities to work on targets that might not be as novel. Frankly, I
Yeah.
mean they're well known, but we have a unique approach to addressing them, and that gives you differentiation. So it's in a Beni is a bit different because that was very novel when we started that program as well. So we have kind of a mix of those, but that's a difference. I'd say like my experience when I was GSK was that we went much more for like first in class, new target, things that
Yeah.
no one had worked on before. And especially with that collaboration with 23andMe that we had back at GSK, we kind of had that opportunity to work with a lot of targets that others couldn't see
Yeah.
in other databases. So that's a difference from the genetic standpoint. Then obviously, as I mentioned already, the size of the organization and kind of working more agile, more faster in many ways, and less regulate, less structure, or maybe not structure, but maybe less red tape or less kind of
Yeah, I
things
hope
that you
so.
have to pass through. Yeah.
Yeah.
So I would say that there are pluses and minuses. Each type of organization have different kind of advantages, but I enjoy this a lot right now. It's been super fun.
Yeah, yeah, that's it's a really interesting insight about it sounds like what you're saying is in a company of Wave size or certainly smaller, like you really need to think about marrying the good target science with the unique capabilities that you have as a
Yeah.
company. As a large organization, you have the benefit if you're Regeneron or GSK or someone like that, you really can discover the new targets. You can get a year or two early compared to everybody else and use that head start to potentially, get ahead in trials or whatever it may be. But you don't, it's very
Yeah.
rare that you can do that in a biotech, right? So you've got to think a little more creatively around what is our platform and how are we different?
I I think so, and I think that's the differentiation for us at least. I mean, there are obviously plenty of smaller biotechs that go for new targets and kind of have their own secret sauce.
Yeah.
It might be based on a technology approach, it might be based on an AI algorithm, but especially if you want to base your discovery in solidly in human genetics as your starting point, then sample size matters. So
Yeah.
so then it's really just like the biggest companies that can play in that, like having unique data that others can't for that reason, I'd say. But
Yeah. You've had a wide range of experiences, not just large pharma, small biotech, but also professor at three different universities,
yeah,
Europe, US. What has driven you to make so many changes? Are you a curious person? Do you just like change?
yeah,
What's the common thread?
yeah, yeah. I think curiosity is probably kind of a, but or just like, yeah, I'm curious. I enjoy. I know that I do a better job if I'm excited about what I do. So I'm very driven by like, having fun, frankly. It might sound
Yeah.
like simple or whatever, but I've always been fortunate enough to be able to make career choices based on things I'm excited about. And that has helped me a lot. And I think going back to why that is, I think it is the curiosity. I kind of just want to learn new things. And throughout my career, even though I spent quite many years in academia, I kind of changed both position, university, but also field. Like I started out with epidemiology, like my PhD is in epidemiology, and then went to human genetics, and then went to functional genomics and molecular biology, and then drug discovery. So like I've been kind of moving around quite much in various areas. So,
What do the human geneticists do that drive epidemiologists crazy like you that you wish everybody understood if they took an epidemiology course?
that's a good question. I never thought about that. I think actually a lot of there's a lot of crosstalk between the disciplines, and it was especially happened around that, like when GWAS started to be introduced, because a lot of those that were kind of in the epidemiology field, like myself originally, maybe they did a bit of like biomarkers and things like that, got interested, and then they kind of transitioned into genetics. They didn't become like proper human
Yeah.
geneticists necessarily, but like genetic epidemiologists. And so that there was a lot of crosstalk, but also kind of probably from the other side, like for those that trained originally with like more traditional, like, linkage studies and things like that, they kind of just based on where the field moved, had to kind of move towards like bigger data sets and GWAS and understanding of handling that. So I think it kind of the fields merged to some extent. It's still different disciplines. You have plenty of epidemiologists that never want to touch anything that has to do with genetics or molecular,
Yeah.
and you also have plenty of geneticists that are like more pure geneticists and don't necessarily want to dabble with GWAS. But I think the fields have merged over time.
Yeah. Yeah, it's one of those interesting combos to me in our field because like when I was looking on the Wave website, for example,
Yeah.
you estimate the patient population sizes in all the areas you go at, and that can be a remarkable piece of contention when it's something genetic, right? Because you look at the epidemiology studies and they have a certain
Hmm.
ascertainment bias. You look at UK Biobank and it's totally different, but not all those people have the disease or have necessarily had enough time to get diagnosed. And it becomes that you can get remarkable ranges, right? In some of these
Yeah.
genetic diseases where you get an order of magnitude difference estimates depending who you ask. And there's not always an easy way to come to an answer. Even things like that, you'd
Exactly.
think we should know how many of these people there are can sometimes give you a very wide range of answers.
Yeah, exactly. Yeah, no, I agree.
As we wrap up here, there was something else that stood out to me that I want to make sure I asked you about, which is my understanding is you've obviously had an incredible professional career, but you also have, I think, managed to maintain a very great balance with family life. I think you spent two years as a full-time dad while your kids were toddlers, which is
Yeah.
something that you don't hear very often from men in our field. And as a father of two young kids, I'm just really curious to hear from you how did that go, and also just what advice do you have for dads more generally that want to make the most out of their career as clearly you have, but also want to be there for their families and have that
Yeah.
part of their life have equal importance.
Yeah, and it's been really important to me. And now, actually, my kids were actually empty nesters as of this summer. So
Wow.
now they're all grown up now. So yeah, I have two kids now. One is a freshman and one is a sophomore in college. Both of them here in California, so that's nice.
Amazing.
So it's kind of new phase in our lives now. But I think thinking back to that period when they were younger and smaller, I for me it was first of all also a cultural thing, and also even kind of a societal or systems thing. So back then, I lived in Sweden, so we have this, maternity and paternity, like so parental leave. So you get quite much parental leave. It's like 1 to 2 years per child,
Yep.
and then you can stretch it out a bit. And so my wife, she's a lawyer, and she's also had a very successful career. So we always like went into that like we're gonna share, and we're gonna like share the burden and do it. Burden, I would rather say joy, really, but like
Yeah.
again, but share how we split that, and we've just been very truthful for that throughout you know our lives. So when they were small, yeah, we I stayed home full time around 2 years in total with them, and and my wife about the same. So we were able to be home with them for the first few years, but then once they started to go to daycare, then at that time I was the one that took most of the responsibilities for like dropping them off, picking them up, and
Yeah.
cooking dinner and stuff like that, because just the place we were at that time in our careers, that was the made most sense for our family. And what I guess the what I did, you always have to make some choices. So I think what I did back then is that I decided not to do clinical work for a period. So I stopped doing clinical work when they were young.
Yeah.
After that, around that time, really. And then I went back to clinical work when they were older again. So I did clinical work again for a few years later in my career, but I took a break from that. So I did only 100% research for that period of time, and then that was much easier. And I was building I was like junior faculty and then senior faculties. I was building a team and all of that, but it was easier to juggle that way. So that was kind of one thing that I made. In terms of like advice, I'm not sure if I have something very profound there, more than again, whatever works in your family. Like just agree to something, but it has been really rewarding for me to be able to take that time and to really kind of prioritize family always. I guess small things for me at least is to avoid working in the weekends. Like
Yeah.
pay attention when the kids are around, and at some point sooner than you think, they're not that interested in you anymore. So then you kind of
Right.
have more time to work again. So
The phone
there's
will still be there.
that.
That's such an
Yeah,
important one of you got to be there the time that you are there, right?
yeah,
Because it's fundamentally limited, and if you're physically present but not mentally or emotionally present, then you might as well be doing something else.
exactly. Yeah,
Yeah. Thanks for sharing that. I
yeah,
would just want to thank you in general for the great discussion we've had here. If people,
yeah.
how can people help you? You're probably hiring, you're probably looking for partners. What's the most useful thing for you for the audience?
Yeah, so if anyone is interested in kind of learning more or in a discussion, just reach out. I am at LinkedIn. So just ping me if you want to kind of discuss something. And yeah, um, nothing more than that, really.
Amazing. Well, thank you. I really appreciate it. And congratulations. 2024 has been a very big year. You joined and you've had this world first as a company. It sounds like 2025 things are not slowing down, picking up a lot of steam.
They're not slowing down. We have a lot of important
Right.
readouts coming up. So it's going to be exciting 2025 as well.
Amazing. Well, thanks everybody for listening,
So,
and we'll see you next time.
Thank you.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.